Welcome to another exciting Dagster deep dive. I hope everybody's staying cool. I know it's really hot, especially for our European audience who may or may not have AC. But yeah, we got a really exciting deep dive plan. And I got to say, as somebody that has primarily worked in the business domain with data, it is like really interesting to hear how folks in the scientific disciplines are thinking about data engineering and really handling the scale involved. So without further ado, I'm gonna kick it over to Keith, the data product manager for engineering at Basecamp Research, and he's going to take us through the really cool things that they've been cooking up over there.
Lovely. Thanks, Alex. So, as Alex just said, I'm a data product manager inside of the engineering org at Basecamp. So I focus on all things data products, which covers assets as we all know them, workflows, so for ad hoc analyses, and also the broader data platform and ecosystem for tooling for our scientists. So taking a step back, at Basecamp, we're building the next generation of foundation models for biology, and we're doing that from the ground up by tackling the data scarcity that is limiting current models and approaches. So over the next half an hour in this deep dive, I'll show how we collect novel biodiversity and we analyze them through a Dagster-powered data platform. By the end of this deep dive, I will hope you'll leave with strategies that you can apply to your own workflows. First, let's go over briefly what the next half an hour will look like. So we'll split things into 4 sections. We'll start off with a little bit of context around biology and AI more broadly, and Basecamp's thesis and our approach to this that we think is quite unique. And then we'll go into what implications our use case specifically has for a data platform, as it's, as Alex mentioned, not quite a modal distribution of what a typical data platform looks like. And finally, for the bulk of the talk, we'll talk about specific challenges and strategies that we used to tackle those challenges. And at the very end, we'll end with a bit of Q&A and having our moderators, Alex and Colton, moderate and read out any clarifications that we have as we go, but also at the very end, longer discussions on what we've done so far. So with that, we'll start by setting the scene and demystifying what is the data wall and why do we care about it for biological problems. So we'll actually start way from the beginning, which is what does data even look like in biology? So for us, one way of looking at biology is through a biomolecular lens. And primarily for that, we care about proteins. So proteins are these nanoscale machines that power all of life, and they're able to do a huge amount of functions that range from things like plastic degradation to making targeted gene edits for therapeutics to things like carbon sequestration in either plants or direct air capture applications. So in order to unlock the potential of biotechnology as a whole, we need to do 2 main things. The first one is to understand and discover proteins from nature. So this is actually finding the sequences themselves, so almost like the bytecode of nature, and understanding how does that correspond into functions that we might care about. And then the second half is to, once we have an understanding of what nature has already done, be able to distill that know-how into the ability to design and optimize these molecular machines. Now, it wouldn't be a talk in 2025 without AI, and this talk is no different. In recent years, there's obviously been incredible progress in AI across various modalities, whether that's vision or language or audio or video as well. And related techniques have also been borrowed across and translated into biology with huge advances in this field. And as a fun fact, last year's Nobel Prize for Chemistry was awarded to work specifically in this area, which is looking at protein structure prediction. So given a sequence of amino acids which encode a protein, what does the 3-dimensional structure look like? And design, protein design, which is given a target function or a target structure, be able to reconstruct and do the backwards mapping back to the original sequence that we might care about. Now, the Nobel Prize for Chemistry is particularly interesting because typically with the Nobel Prize, it's awarded decades after the initial work. Nobel Prizes are awarded for things that have transformational impacts to an entire field. So this really shows the impact that AI-guided protein design has already had in the wider field, given that it's happening literally 3 or 4 years after the initial work has been done. So the leading approach to this very broad class of many different types of problems is training large foundation models. And this is no different from the ChatGPTs of the world. So you're training with large unscaled, large unlabeled data and using these models and fine-tuning them to specific tasks that you might care about for prediction and design. And to grossly simplify, AI can be broken down into three ingredients, if you will. The recipe for AI is, first of all, scaling compute infrastructure, the GPUs and the clusters that they become a part of. There's the second part, which is improving algorithms and improving the efficiency by which AI algorithms are able to learn from data. And the third part, obviously, is gathering and improving and curating the data sets that feed into these models. And in particular, our thesis is that these foundation models, for biology at least, data is the last part that we think there's lots of low-hanging fruit to be gained. And it's the part that we're primarily focused on. And when it comes to data in the wider AI landscape, the models that we all know and love, right? They're pre-trained on web scale data, which is mind-boggling to think about. But flipping back to biology, we're much earlier in that maturity scale of how much data do we have and the breadth of the data that we have access to. So this is both in terms of the scale of the data and its rate of growth over time, as well as the data diversity and the quality in those data sets. And one particularly interesting problem, if you're from a data science background, is the class imbalance. So large proportions of these data are coming from very few species, so very few taxonomical groups, and very few regions globally. And naturally, if your data doesn't reflect the underlying distribution that you care about, which is all of nature, your downstream metrics are going to be impacted. As our founder described it, it would be like trying to train GPT-5, but instead of the whole internet, you're just including like the Bible and all of its translations and analyses. That's probably still quite a lot of data, but it's by no means nearly enough in order to get a generally intelligent AI. So our approach is to avoid or not use public databases, but to build our own data from the ground up. And what that looks like is this. So our database is built through on-the-ground exploration in some of the world's most extreme and biodiverse environments. And the photos that you see here aren't stock photos. These are photos of our field team and our partners collecting samples in field expeditions. Our field team and the partners that they work with, they'll sample from hot springs to polar ice shelves. They'll sample, I think there you can see a shipwreck as well as rainforests. At a very high level, the data flow looks like we have our global research team. They first acquire all the permits required in order to sample from these various locations, and then they will work with partners to take physical samples from the environment. So this might be a bit of soil, it might be some water. Next, these samples are processed based off of the sample. We do different processing techniques to them, and we extract the DNA from the microbes that are contained inside of these samples. And then finally, we use high throughput next generation sequencing to digitize this genomic information. And then that's where all the data platform work starts. So at the end, each sample being processed in physical reality, we have a large number of DNA reads that we then piece back together like a puzzle piece. And then we try to run annotation tools to extract information of what are the proteins that these microbes are producing in various different environments. So we're building scaled data for foundation models. And in order to achieve this scale, we're focused on building trusted relationships around the globe. And to do, we embed benefit sharing, legal clarity, and long-term collaborations with our partners around the world. So our partners include researchers, local communities, as well as governments. So they can be either local governments or national parks or even at the national level. So in each of these steps, we are embedding, as mentioned, these benefit sharing back to our partners so that we can make sure the world's largest biological data set grows inclusively, ethically, but also like sustainably with all our partners over time. So recently we've released a little bit of a sneak peek into what we've been doing over the last 5 years, which is culminating into initial preprint that looks at our data collection work. And this results in the largest biological sequence database. And it's worth calling out that there are all these high-level metrics around how much is in that database. But the really interesting part is, to me at least, it's not just the raw data volume, it's the diversity that's contained inside of it, whether you're looking at it from a protein diversity angle or from a taxonomical angle. So to recap the first section in brief, as a company, Basecamp, we built a supply chain for gathering novel biological data, as particularly sequence data inside of DNA. And this has allowed us to, well, from that, we're working with a global supply chain to build that input. And then this has resulted in our ability to sustain and scale data growth over time, especially over the last year. And using this data, our aim is to teach AI to design biology. And at the end of the day, once we've achieved that, the goal is to realize the potential of biotechnology overall, whether that's curing diseases or developing more sustainable industrial processes or making crops that are more resilient and hardy as we need them to. So I'm going to pause there. Alex, any clarifying questions on that front?
Yeah, we actually had a question from Arturo, which is, so one of the first successful projects in this space, AlphaFold, used deep learning at the time as the basis for predicting 3D protein structure from amino acid sequences for
Yep.
protein research, design, etc. But to better understand Basecamp research, what's the equivalent statement for you guys? And where does this approach scope out of the two projects overlap and differ?
Yeah. AlphaFold naturally is the really landmark piece of work from DeepMind on protein structure prediction. We've shown that using a very similar technique in the model itself almost has you can think of it as a RAG component where it first fetches proteins that are similar to what you're trying to predict and then puts them all together in what's called a multiple sequence alignment. So this is basically just like having similar related protein sequences all put together, and then that's what gets fed into the model. We were able to show that using our additional data, just by improving effectively the quote unquote RAG part, we're able to get better predictions at the end. So that's just an early way of showing how this data is beneficial in these downstream tasks. More broadly, our data we're looking at foundation models in a variety of lenses. I don't think I'm particularly at liberty to share this yet, but we're looking at them in different modalities for looking at our data over time.
Awesome. And I actually had a question. When it comes to these proteins, I believe you said that for like these models, the microbes, as far as species, are the ones that are primarily used. Is that just a function of the ethics around using those species, or do they provide more utility towards like the downstream applications versus like animals, plants, fungus, those types?
Yeah, I think there's kind of two ways you can look at the problem. One kind of basic thing is just that there's so much more microbial diversity than there is in higher organisms. So even though there's more complexity in each organism, the sheer variety of the strains that are available in like a teaspoon of soil is immense. And you can think of it as there are these really parallel experiments where each microbial strain is doing lots, it's trying all of the things and interacting with its neighbors, and the amount of protein diversity just in prokaryotes is like really large. So this gives us a really relatively economic way of sampling a large space in protein design effectively.
Awesome. Fascinating. Yeah,
Cool, cool.
we're good on questions here, so we can plow ahead.
Sounds good. So in the next section, we're going to talk a little bit what this high-level problem space means for a data platform. And these are things that we actually dug up from around a year ago, our notes when we were looking at revamping our data stack and deciding on what we want to build it off of. So the first thing that we call out is what we literally just talked about, which is the increasing data volumes. As a metric, last year we 10x our data, which meant that our orchestration and infrastructure needed to be upgraded. Previously, when we were looking at dozens or hundreds of samples, it's still feasible to do fairly ad hoc techniques. But as we scaled up the amount of input data that we were putting in, we needed to automate all parts of the operations of getting this data from its raw, uncurated form into a form that our computational biologists and protein scientists are able to consume. And the second part is more on the people and process and the personas that are involved in the platform. So at Basecamp, we're always working at the intersection of different data and software fields. And I think this kind of encapsulates the different tooling that each of these particular domains might care to include. And while these specific personas, I think, are quite specific to us, I think the takeaway from here is to note that you're likely also going to have very similar problems in any company, whether that's looking at finance versus advertising versus supply chain. Each of these domain experts will have their own set of preferred tooling. And the important part is how do you unify all those inside of one data platform so that all of these people can do their data work in a consistent and replicable manner. So for us, we're starting off with the biological folks. So we have the bioinformaticians, and what they're primarily interested in is what can we do to extract more value from our raw data? That can look like developing new annotation tools. It could be looking at evaluating off-the-shelf tools. And then once they figure that part out and benchmark them against real data, scaling that out to applying it across our entire data set. And we've got the second persona, which is looking at software and analytics engineers. So for this persona, that includes a lot of the data engineering team. And for them, we care about building large-scale performance data assets, whether that's in a data warehouse as a table, or whether that's as a workflow or a CLI that allows people to retrieve or search across our large database. Given that we have fairly large, unwieldy data sets, we need ways to be able to index on top of them and have biologically relevant ways of searching across and filtering that data. And then finally, we have deep learning folks, or the AI folks, looking at deploying ML models, but also curating training data so that they can train the next generation of models. And then as we work on, as we briefly mentioned earlier, sort of more foundation models that are less task specific, building out evaluation pipelines that allow us to automate more of the grunt work associated with iterating on models and data. So if you're interested in this idea, I'd encourage you to take a look at this blog post from Dijkstra last year. In this, they make the case for a variety of things, but one thing that really resonated with us was the point on data platform heterogeneity and the fact that even though you have really diverse use cases and personas and tools, you should still have them under one interoperable lineage graph and one interoperable orchestrator. And we'll also, for that last point, talk a little bit about that later in one of the problems that we had previously before moving to this approach. But to recap in brief, we have really rapidly growing input data. So our data platform is growing in terms of the amount of raw data that we're trying to process. We're also growing in terms of within the data platform, the number of use cases that we want to do, so the types of analyses that we want to do, as well as the sheer number of analyses that we want to do. And also from the third axis, which is the number of people that we want contributing to this platform. And on that third point, we want to make sure that whatever the tooling folks want to bring, they have a way to do that ergonomically and idiomatically inside of our data platform, whether that's bioinformatics or analytics or ML engineering. Cool. Alex, any questions?
Yeah, we had a few people asking about like what is your architecture and the different like tools that you use to accomplish all this. Now, I imagine we're going to cover this in the next section, but is to tee up that conversation, are there any tools that is particularly helpful in the bioinformatics domain that you've
Yeah.
trained for this?
So bioinformatics is a very interesting one because that's fairly new to me as well. I would say it has fairly different idiomatic ways of developing pipelines compared to what we might be more familiar with if you're from a data science Python background. It's a environment that's a lot more CLI focused. So the individual steps tend to be orchestrated in terms of the atomic unit of a CLI invocation. And then the orchestrators are built around that abstraction instead of something that you might see in a DVC or a flight or weights and biases. So I would say that's the main thing that we had to cater for, which I think we'll dive into a bit deeper as well. The other thing that I think is useful to call out is also that we use DBT extensively. So I'm sure many of you in the audience are also folks that are looking at integrating their analytics workflows and data warehousing as part of a wider data platform. We also have very similar needs because ultimately we want data sets that are queryable and that we can join and do additional things to. So we still have the requirements to do that, albeit on a fairly large scale. So one of the main points that we want to talk through is also how we use DBT.
And then we had another question from Juha. And as it relates to having these different personas using your data platform, do you separate out your project for code locations by these personas, or is that not the strategy you use?
Yeah, actually, that's one of the first things we're going to talk about next. But to give a bit of a teaser, we do use code locations for particularly GPU related things in particular, but we also need, because of the bioinformatics workflows, we have some requirements that mean that code locations don't really scale particularly well to that. But for code locations, we do use them for, we have, for example, a GPU-based code location for running inference on Hugging-based models. We have another one that's looking at GPU, particularly for visualizations and large-scale data frame processing. So I would say we have a few separate code locations for core workflows that have perhaps incompatible dependencies, but we have an additional framework that we've built on top of Dagster to manage the wide variety of tools that we care to install from a bioinformatics perspective.
Amazing.
Lovely. All right. I think that kind of really tees us up for the next section, which is the only way to begin is by beginning. And for us, that's quite literal. It's how do you install all these bioinformatics tools? And unfortunately for us, as a software engineer, you can't just UV install all of these tools. And because we're primarily bio-based data platform, we need to be able to handle all the tools that a bioinformatician would want to install and run. First thing to note, as I briefly touched upon earlier, is that there's loads and loads of tools. And the typical way or idiomatically way of installing these tools inside of a bioinformatics pipeline is to have each step of the pipeline have its own isolated environment or just install that particular tool. For us, that would mean literally dozens, if not hundreds, of code locations, which doesn't really scale particularly well. So to tackle that, we had to come up with a lightweight way of defining reusable or almost disposable virtual environments. The next one is that the installation channels are also very, very varied. So you have the things that are typically installable via pip or UV or via Conda or BioConda channels. You have things that are perhaps system packages, but also things that our internal users might want to fork a code base from GitHub and then make some additional patches to, but also install another pre-built binary. So there's not one single install path that covers the gamut of what we care about. And as mentioned, there's a really long tail of customizations, whether that's applying patches to code bases, whether it's custom tweaks, whether it's installing CUDA drivers. There's lots of additional things that we need to care about when it comes to installing these tools, which kind of leads to this problem that we have of we want to unify everything under one roof, but that also means that we need to be able to install lots of tools really quickly and make sure that developer experiences not too hampered by the use cases that we want to support. So the first strategy that we took for this is to treat containers as an asset and then build out our internal SDK for installing tools under that assumption. So we built out a little internal SDK for building container images based off of some kind of spec. So the first is to support publicly available container repositories. The second is to have a fast path for building containers that just install dependencies from Python, whether that's via pip or Bioconda. And around 80% of our use cases for bioinformatics tools specifically follow this installation pattern. And then finally, to fully capture the tail end of customizations that we need to be able to support, we also support installing directly from an internal image repo, so just post it on ECR for us, or having a inline Docker file inside of our data platform repo. One thing to call out for these containers as assets is that they're intended to be used as like single line entry points, where you're invoking a asset that has a container and a CLI command as an input, and then that's almost treating it as one function that you're invoking. We don't have full Dagster runtimes. We just use them to run specific tools as part of a wider pipeline. So this also leads into a broader problem space, which is how do you actually develop these pipelines? Installing the tools is one step of the challenge, but the second step, if you step a little bit further back, is all of the different interacting components that you would have inside of your production data platform. So for us, that's looking at using developer containers, and specifically its Docker Compose setting. So developer containers are a feature inside of VS Code and its associated friends, so all of its forks. And it allows you to define a specification for reusable container that you use to host your repo as you develop so that you have all the dependencies built in, but it also allows you to spin up associated services using Docker Compose. And we use that to have a local environment that matches as closely as possible our production environment so that we can test integrations between, let's say, Dagster's runtime interacting with S3 or Dagster interacting with our ClickHouse instance. So as you can see here, we have what we were earlier talking about, which is how do we install different tools? We don't run them inside of the dev container or the Dagster environment or code location immediately. We have a resource-based abstraction that then builds these tool-specific images and invokes them with container runner, which is another resource. And then we have all the auxiliary services. So let's say we use Minio for a S3-like interface. We use ClickHouse and our DBT resource to connect to it so that we can test publishing things into our data warehouse and also interacting with the data warehouse under DBT. And then we also have a Postgres connector for any transactional workloads that we might care about. So this gives us a really nice way of encapsulating all the things you need in order to start developing on the platform. And it means that we can streamline the onboarding process and you don't have this, it works on my machine kind of problem, because hopefully this is a fully specified way of defining what works on my machine means. So with that, I'll pause a little bit on this current theme. I've structured these takeaway slides as a two-panel slide. So on the left, we have a message takeaway for a scientist, and on the right, we have to the engineers. But for nomenclature's purposes, I think when you see two scientists, what I want you to think of is any domain expert that consumes or analyzes data and wants to share their findings. And for the engineers, I would say that's folks more on the data engineer or data platform engineer spectrum. So people that are looking at building tools for others or building frameworks for other people in their team to use. I personally would see these almost as a spectrum, and that as people get more senior, they like work on responsibilities on both sides. But with that, I'll stop with the aside. For the scientists, I would say that the key takeaway here from this section is that if you can treat your definition of done as someone else being able to reproduce your findings without your assistance, that's a lot more sustainable going forwards over time. And container images is a very useful tool to be able to package up your code in a way that's very reproducible. And the person that's reducing it could be a platform engineer that's trying to productionize your code. It could be another person in your team, or it could be you, right? It could be you a year and a half down the line. I know personally that I can find it hard to reproduce code that I've written myself. So consider it. So as your team grows, this becomes a bigger and bigger problem. And for the engineer, I would say that the takeaway here is that tooling for your data practitioners needs to capture those specific needs. And the best way to do that, I think, is for us, dogfooding your internal APIs and building pipelines on it to really understand like which parts of the API feels funky. And that's been really helpful for us. Now, the next challenge that we have is developing and maintaining complex data pipelines, again, primarily through the lens of bioinformatics. I'm going to talk a little bit about challenges that we have with traditional tooling in that space. So first, there's lots of different file formats and data formats, and these are all bioinformatics specific, and they have many quirks and edge cases. Some of them are human readable, some of them are binary only. Additionally, because the file is the core data abstraction for these pipelines, all of these tools expect raw paths and directories instead of typed objects, which makes understanding pipelines potentially a lot more difficult because the variable itself is just a path, but you don't really know what's to expect inside of these paths unless it's very clearly defined inside the code. And the third one is that schemas inside of this paradigm can change quite easily. And we found, at least I've been burned a few times by this, where schema that you expect, let's say from a TSV file that's from a given annotation tool, can change as you change tool versions or as you change the specific options that you pass into the CLI. So just to give a flavor of what this looks like, the pre-existing status quo before we looked at revamping this was built off of open source pipelines, and this involved, if I recall correctly, our orchestrator invoking bash scripts that were wrapping Perl scripts that were wrapping tools that themselves were wrapping other tools, which meant that there were loads of conflicting dependencies, but also understanding the pipeline itself became really non-trivial because there were many layers of indirection and abstraction that weren't cleanly putting all of the business logic in one place. So for us, the overarching strategy and what we've tried to popularize in our data engineering as well as bioinformatics team is an internal SDK that helps us express our intent through types. And the first thing to start with is just using file types. So instead of having a string to represent a data object, we use a type that's based off of the specific data that's contained inside of them. For example, for protein sequences or DNA sequences, we would have a specific type that is specific to that, or for a protein structure, or for a Parquet file for tabular data. So all of those types get encoded inside of that class. And these can compose into how we actually use those classes, which is CLI commands. So we're building on top of the previous, and we're making it explicit. This command expects this file type as an input, and it will output this file type, even though the underlying command just ends up hydrating it to maybe an output option flag. By encoding it in our pipelines, we're able to express intent as to, oh, this asset after this running the CLI command, we expect the output to be a file type A, and then that file type gets propagated down into in between assets. So then as you're building on top of these pipelines, you're able to inspect what you expect from a given step inside of that pipeline. And to build on that, tabular schemas is another thing that we particularly care about. We use files as the natural interface, and on top of that, we have the definition of what does a tabular file mean, and we've enforced it so that we have specific schemas associated with those files as well. So we define a Parquet file as our commonly used interface for tabular data, but we also require the subclass to encode the schema itself so that we can do data validation on read as well as on write. So we use Pandera, Polars, and Parquet for this, which means that you have this nice Pydantic-like interface for defining a tabular schema, and you can add additional things like column level validations for strings, let's say, if they have an enum value that you expect it to adhere to. One other nice benefit of this, if you need a way to sell this to your team, is that types are really helpful for AI agents. They're helpful for human agents, but they're also very helpful for AI agents because typing code allows the language server protocol or type checkers to catch basic code errors. And this allows, in most modern coding agents, to feed that information back to the coding agent so that they can make that change without you having to intervene. So it's able to self-correct and be autonomous for longer, which makes you more productive with coding agents. So the second major benefit for us by defining all of these types and structs is it allows us to build platform features like custom metadata, which we expose in the Dagster UI. So based off of the file type that we just talked about, let's say we have a media file like a SVG or PNG, we can, for example, display a preview of that image directly into Dagster's UI by dumping the binary contents into a markdown entry. Bit of a hack, but it works really well. With things like Parquet files, we can, for example, display both the tabular structure, so all the columns, the descriptions, the types, as well as it ties into the Dagster's column lineage features. We can also do things like display a preview of the data or display statistics around the data. And this carries on to biospecific formats like sequence files. We can use domain-specific profiling tools to extract summary statistics that we care about. And we found this is really helpful because it gives more confidence for people that are newly exposed to the platform to understand what's going on, because you can see, okay, this asset corresponds to this file. I know what that file is. These summary stats also make sense to me. It makes it a lot more approachable to explain what's going on in an asset instead of just having a really long doc block, doc string that gets rendered out. And it's also really helpful for debugging what's happening in a pipeline, because it means that you don't need to go into the underlying file. Let's say you had an issue somewhere in a long pipeline, you can just look at the previews of the upstreams, and then sometimes that's enough to tell you what's wrong. So it's also really helpful from an observability perspective. So the takeaway on this side, for the scientists, expressive code is a lot nicer than docs. And to be honest, most of us don't write docs anyways, and they're always out of date. So if you're able to encode what you expect for, let's say, data, that allows the computer to tell you something unexpected, and also importantly, gives you context to whoever's reading the code, whether that's you 2 months down the line or whether that's someone else joining your team. So documenting your intent in code in terms of typing your data and what your expected outputs are, while it seems like a bit of a tedious thing in the beginning, it becomes really helpful later on as your code base scales to the point where you can't fit it all in your head or your agent's context memory. For engineers, I would say that your point of leverage is developing platform features, and these platform features ideally are orthogonal to the business logic of your pipelines. The things that are in your data platform obviously include the applications, but for you, developing features that make it easier to develop these pipelines or run these pipelines or debug these pipelines is very important. But for them to be useful, they need to be tailored to your domain. And I would encourage you to speak to your users and figure out what they are looking for in a platform to make it easier for them to develop. That can be things like investing in the data level observability, or it could be looking more at the infrastructure side of things and seeing what resources a given job is consuming and understanding that. But yeah, that is, I think, the main takeaway from our past year of working on a data platform for this. Cool. And the third theme that I think we want to talk a little bit about is, as mentioned earlier, the data warehousing and operations complexity. And the first thing to call out is like what this kind of looked like before. And our initial sort of legacy data flow involved, we had some kind of orchestration on the cloud that was running all of these, as I mentioned earlier, the bash scripts wrapped in Perl scripts and things like that. And then these pipelines output files in S3 under some kind of structured, semi-structured path that we would then infer. And then we had some custom data ingest, and then that was going into Postgres, which was our data warehouse, which we then wrangle the data using dbt on. The main thing that was really challenging for us here was this central part of there were effectively two orchestrators, but no way to communicate across them. And this meant that it was hard to understand what the pipeline itself was. And let's say like I had a table at the very end that I cared to give my end users. It's hard to explain to them what part of that code, how that data was generated. Like all they would be able to see is the parsing logic of getting that like raw blob in S3, but that wouldn't really tell them what settings were used to generate that file, what's upstream of that. So there's no lineage that goes all the way across. And the second part of that is because they're separate orchestrators, you don't have observability of what state the entire project is in. If there's a missing sample somewhere in one of these tables, there's no way to know at what point something failed. And this became very challenging because it you started having data issues that were very silent, and just having missing data was very hard to triage because we would keep getting new data coming in at the beginning, but at some point it would drop out, and then we wouldn't really know when we would expect data either because sometimes things would just fail silently. The other interesting thing is because the orchestration did not really have state in the upstream cloud-based orchestrator, every time we added a new analysis or a new asset, we had to have two separate processes where we had the way to update our prod pipeline, which is what all new samples would be run on, but then we also had to backfill all of our old samples. And this was very tedious, first of all, but also very error prone. And it meant that you had this additional way of introducing desync errors. And the annoying thing is it's also really hard to rerun and figure out what things need to be rerun. Let's say we upgraded a tool. There was no way to know which assets were now desynced because the data generated process has been updated. There's no versioning in that sense. And then the third one is on the other side, running dbt itself becomes really hard because the orchestration on that side doesn't really have a good way of understanding within a table which partition has been materialized. So for us, the natural way of defining our partitions is effectively per sample. So like a physical sample corresponds to like a group of rows inside of a table. But within dbt, the incremental macros that we had access to when we were working with this was primarily time-based. And this is totally fine for really end stage tables at the very end of your pipeline. But once you have joins across different incremental macros, it becomes very hard to reason about which are the partitions that you actually need to update over time. And the current end state now is we have Dagster embedded throughout the entire pipeline, where we have the bioinformatics pipelines are orchestrated under Dagster. The way that we then pipe that data into ClickHouse, which is our current data warehouse, is also tracked within Dagster as well. And we've built out custom scaffolding to enable that uploaded data into S3 so that it's available to ClickHouse. And then we then further track the dbt materializations inside Dagster as well. So as mentioned, we have the dynamic partitions tracking across assets, which allows us to have a lot more visibility in terms of what things are currently materialized, which things have failed. With dbt specifically, it was also very important for us to be able to partition these tables because this allows us to have partition level materializations. So for that, we use dbt vars, and there's an example inside of the docs in Dagster of how to use this, which we found were very helpful. Effectively, it allows your dbt assets to have an associated partitions definition, and then all you need to do is encode how that partitions within the context gets passed into the dbt invocation. And then that allows you to select partitions instead of running fairly crude ways of incremental materialization. And then for us, the third thing that is really important is more of the dbt ClickHouse option, but then the incremental strategy of using insert overwrites based off of a partitioning scheme allows you to do atomic swaps of the data so that you don't have partial data as in case dbt run fails midway through. And then another thing that was particularly interesting for us was based off of this existing setup, it also meant that we can now do things like point-in-time snapshots. And this is particularly important in the case of trying to get a global view across our entire data set. For example, when we're trying to describe what we have in our data set in our preprint. So as mentioned earlier, most of our data pipelines are partitioned by sample, but occasionally what we care about is not by sample, but almost like a whole database snapshot and understanding for example, what is the redundancy that we have across all of our samples. So in order to do that, need some way of viewing a snapshot, whatever that is. For us, we did that through the source tables in ClickHouse. So our source tables are all stored inside of S3, and we're effectively defining a virtual table based off of some path prefix, and ClickHouse allows us to do that. So we use that as our common interface for defining snapshots later on. This allows us to do effectively time travel and figure out what state that source table was at a given point in time. And we can generate a point in time snapshot that describes the state. And we can make this efficient because we can do this query partition by partition. Each given partition can only have one state at a given point in time. So we don't need to do it at a row level. We can do it at partition level. And then with this, we can apply a snapshot query. Typically, all of our query templates are all partitioned as well. So then we can fan those out and then do the point in time query with joins and whatever across many partitions at the same time. And then we can collect all those results and then get back an output. Now, one thing to note is that while this made sense for us to do at the time, it probably doesn't make sense right now because there are better options for time travel. At the time that we were investigating how to do this for our use case, table formats like object-backed table formats like Iceberg didn't have great support, for at least in the libraries that we cared to use to interface with. And we thought that this was an easier way to get started with that, and we can migrate to something a bit more robust later on. But these days, I think it's very sensible to get started directly with something like an Iceberg and related catalogs because they're built to have this point-in-time querying ability. So this is what that code looks like. We define a template of SQL queries. This is basically just a Jinja templated SQL query. And then we have on the right some resources that define how we can take that query template and then take a snapshot across. So under the hood, it does all those individual steps. But for a user, they only need to define a query template following a set of guidelines. So for us, we're back to the takeaways. For scientists, I would say that thinking about not just the core analyses, but also how it can be turned into a DBT table or a data set that is easily shared and easily rerun and versioned across both the data that's going in as well as the code that's going in is very helpful. And for us, our users got into the habit of defining these staging tables that they can then define hex apps and dashboards on, which is particularly helpful because then their endpoint becomes more like, how can I communicate these results to end stakeholders instead of just, I have this analysis running locally. I think that by going that extra mile, you unlock things like allowing other people to iterate quickly in your dataset instead of having to ask for CSV in Slack. It allows you to automate refreshing this data so that you don't need to rerun the analysis each week or quarter when new data comes in. So I would encourage you to speak to your platform team at least and understand like what resources they have available in order to go the whole mile. And for engineers, I think the interesting takeaway here is thinking in partitions and how your data sets can be run incrementally if possible. And that's particularly important as data scales. And obviously, we're not at giant web scale data yet, but this has been really effective for us to manage the data scales that we have in this intermediate range and being able to iterate more quickly on the data assets and pipelines that we're trying to develop. So with that, I've got a little bit of a TLDR on the different themes and challenges that we had and the corresponding findings. And of course, I couldn't have done this without the team that has contributed to the data platform. So big thank you to them. If you're interested in our work at Basecamp, I'd encourage you to follow us on LinkedIn. We're hiring for various roles, including senior platform engineer, a product technical program manager, head of AI, many others. So I'd encourage you guys to check us out as well. Great, thank you.
Yeah,
Maybe
so
if you have any more questions.
yeah, we have a few questions. The first one is around like how do the downstream stakeholders, whether they're a scientist or a data analyst, how do they know what is the right like semantically versioned asset to use. Like I imagine your custom metadata as it relates to assets helps a lot with that. Does
Yeah.
that create like a sort of self-service functionality for those folks?
Yeah. I would say we're fairly early in our journey for that at the moment. We're trying to get the primary core strategic use cases up and running first, but we're definitely very keen on using more of the catalog functionality and exposing the catalog to more end users. And this has been a great motivation for me to write doc strings effectively for each of the assets that I'm working on. Because in the back of my head, I want this to be something that I can share directly to users. That's been quite helpful. I think the main thing that we have currently is the end consumer assets are effectively most of the time DBT assets. So then we're incorporating the DBT level metadata, table level and column level metadata, and a lot of that is just exposed through the data warehouse effectively. So have hex sync through all of that column lineage so that people can search across it and query like which table has the column that I care about. Naturally, there's still challenges with that because it doesn't show all the lineage, but at some point we're looking to move towards using more of the observability tooling in Dagster as well.
Amazing. And then we have time for one more question. And this one relates to like how the project went as you were developing base data. Was there any
Mm.
particular moments along your journey that really accelerated the development of the entire project?
Yeah, I would say we were looking at this because we had some additional engineering capacity at the point in time. And then as it became clear that we were going to do this very big data ramp, the priority rapidly increased because the number of samples that are like bioinformaticians needed to babysit in the old system meant that it was very clear to them that if they needed to 10x the number of samples, that's all they would be doing all day. So it became very easy to get buy-in into sort of how can we do this more effectively? How can we have the tooling so that over time this becomes things that we can have hooks to build in additional tooling that they would care to include scaling resources based off of the data that we're doing sort of end-to-end lineage and things like that. So I think I would say the large part in being able to gather the engineering and sort of stakeholder engagement for this is it was a strategic priority for us as a business to be able to actually analyze that amount of data. Yeah.
Fantastic. We're going to have to cut it here. I want to thank everybody that attended for giving us such a great session and great engagement and questions. And I want to thank Keith for putting together this banger presentation and really distilling a lot of this like really complex engineering and scientific content into more digestible material. So I really appreciate that. And
Lovely. Thank you, Alex.
yeah, have a great day, everybody.
Thanks, guys. Bye bye.